In September of 2020, the first new variant strain of SARS CoV-2 virus, which is a virus that causes COVID-19, was found in the United Kingdom. Subsequently, new strains have been found in South Africa and in Brazil. Why is this virus mutating? How do we know it's mutating? And what does that mean for all of us? In today's episode of A Taste of Medicine, that's what we're going to cover. Let's go. Fancy new graphic and everything. Thank you, Fiverr. Hello again, I'm Michael Lawson. I am a hospitalist physician and board-certified internal medicine doctor in Minneapolis, Minnesota. As always, any opinions shared on this channel or in this video are those of myself and not that of my employer. The other disclaimer we just have to talk about is this is the most rapidly changing aspect of the pandemic, of these new mutations and these new strains. As a result of that, a lot of the papers we're going to be covering have not had the chance to be peer-reviewed as of yet. Peer-reviewed is important. What that allows is other experts within the same field to read the study and determine whether there are any serious flaws in the way the study was performed. And if they do find those serious flaws, then it would not be published in any major medical journal. SARS CoV-2 is a single-stranded enveloped RNA virus. The reason that that's important to note is that RNA viruses tend to mutate more quickly than other types of viruses. There are multiple reasons why viruses change over time, but today we're going to stick with one aspect, which is mutation. Mutation happens when part of the genetic code of the virus itself is miscopied. When the virus enters your cell, it uses your own cellular machinery to make a lot of copies of itself. And in that process, there's a chance that something could go wrong. And while it's trying to copy itself, an error is made. There are different types of mutations as well. Sometimes additional genetic material is encoded by mistake. Sometimes parts of the genetic material that are supposed to be copied are left out or deleted. And other times, different amino acids take each other's spots. And as we mentioned, because SARS-CoV-2 is an RNA virus, it is likely more prone to having these errors when it's copying itself than other different types of virus, specifically DNA viruses. And this is because a DNA virus uses an enzyme called DNA polymerase as kind of an autocorrect. This cellular form of autocorrect helps prevent any errors from happening when a DNA virus is replicating itself. But RNA viruses use a different enzyme, RNA polymerase, hence the name. And RNA polymerase doesn't have the capacity to autocorrect or proofread the copies that the virus is making of itself. And as a result, mutations are generally more likely in RNA viruses. In the description, I'll link to a very nice article kind of outlining the general principles of this by Khan Academy. I'll also link in the description to a podcast on this very topic by The Atlantic. In the podcast, they talk to Dr. Vineet Minatri. He's a PhD in immunology and microbiology. He works at the Galveston National Laboratory. He has been, in his own words, doing research on coronaviruses since before it was cool to do, and is able to provide a lot of useful context to the three major strains of mutations that we've seen so far. As I mentioned, the first major strain was found in September in the United Kingdom. Now, it's possible that this truly was the first major strain that mutated in the world. But it's also possible that it was first found because the United Kingdom is leading the world in terms of genomic sequencing. As of December 29th, there had been a total of 275,000 samples worldwide that had undergone genomic sequencing of the SARS-CoV-2 virus. And almost 50% of that happened within the United Kingdom alone. But we'll talk more about that in a little bit. So it's possible that the UK variant is the first mutant strain in the world. But it's also possible that it's just the first one we found, because that's where we were looking the hardest. As I'm sure many of you have heard, It's believed that this strain is significantly more transmissible, oftentimes quoted as being 70% more likely to go from one person to the next than the original strain of SARS CoV-2. And part of the reason that researchers believe that to be true is because it was first found in September and is now the major strain in the United Kingdom. Nearly all of the mutations that have been identified revolve around the spike protein. And if you've seen the previous episodes of A Taste of Medicine, you know the spike protein is really important. The spike protein is the spiky protein that sits at the surface of the virus. And this is the way that it gains access to your cells. So the best analogy I've heard is to think about the spike protein rather than as a claw or as a needle, as a key. And as that key mutates, some of those keys are going to be better at accessing your cells, and some are going to be worse. This is the crux of evolution. The interesting thing when it comes to virology or immunology is that because the life cycle of a virus or a bacteria is significantly shorter than that of a human, you see rapid evolution over what we perceive to be small periods of time. So because that life cycle is much faster, we're going to see more mutations in a shorter period of time, and any of those that grant any sort of competitive advantage to one strain over the other will be more likely to be successful. And the way a virus is successful is simply by going from host to host and replicating. There are animal studies going on as we speak to try and get a sense of how much better this key or this spike protein is in these other variants. Is it twice as good at getting in the cell? Is it 10 times as good as getting in the cell? We don't know right now. When we talk about transmission, that's only talking about how easily it goes from one person to the next. How likely are you to catch it from someone that you're around? It doesn't mean that that same strain is more lethal to a given individual. And as Dr. Manatri points out in this podcast, there's often an inverse relationship with the severity of disease and how easily it spreads. Now that might seem weird, but if you think about it, it's pretty straightforward. Take Ebola. Well, don't actually take it. It's a horrible disease. Ebola kills around half the people who contract it. And if you're catching Ebola, you are going to be extremely sick. But in some ways, that's a saving grace, because the worse you feel, the less likely you're going to be out and about spreading it to other people. And on the other end of the spectrum, take the common cold. No one dies of the common cold. It'll make you feel pretty crappy for a couple of days at worst. But pre-pandemic, a lot of people still went to work plenty sick. Very likely spreading it to their coworkers and people on the bus or on metro. So while a common cold will spread very effectively, it's not going to kill anyone. And SARS CoV-2 is in the middle of those, right? It's the perfect storm. It certainly causes plenty of death and destruction to a good chunk of people who experience it. But there's also this larger chunk of asymptomatic carriers or minimally symptomatic carriers who are spreading it to lots of people. So how are vaccines impacted by these strains? That is the million-dollar question. We just don't know the answer to it yet. There is one study, again, not peer-reviewed as of yet, that was published online January 7th. In this study, researchers at the Galveston laboratory, along with some researchers from Pfizer, so the researchers took a strain of the SARS CoV-2 virus, which had a singular mutation to the spike protein that is being seen in both the UK variant strain and in the South African strain. They then took sera from 20 people who had already completed the Pfizer vaccine series. Unfortunately, they found that there was equivalent neutralizing titers. So that's promising, and that's exciting. But it should be noted that both the UK and South African variants have other mutations too. We don't know for certain if the results would have been as positive comparing it to the predominant strain in the UK and the predominant strain in South Africa. The spike protein is around 1,300 amino acids long. So it's certainly possible that one singular mutation could have really big impacts on how easy it transmits from person to person, or how severe the disease is, or how effective the vaccines are. But the antibody response is to multiple portions of the spike protein. Think of your immune system as kind of using shotguns rather than sniper rifles. The other thing to know about likelihood of mutation within a virus like this is that the virus is replicating a lot within just a singular host. A study published in The Lancet showed that the average viral load in the handful of patients that they surveyed at the time was around 80,000 copies per milliliter of blood. So it may seem like this virus is mutating a lot in a short period of time, but you have to keep in mind that each person who's infected with SARS CoV-2 has the potential to have an incredible amount of virus in their body, and each of those virus particles are going to be trying to replicate. And that's just in one person. At the time of this filming, the global counts are north of 93 million infections. Here's another article that was published on January 4th online. Again, this one has not been peer-reviewed as of yet. They took the plasma from the 17 patients who have recovered from SARS CoV-2 and have antibodies to the virus itself. They then compared how effective that plasma was on various different mutations around the spike protein. They did find a particular set of mutations that related to those antibodies being 10 times less effective at neutralizing the virus than in other strains. Again, this is one study of extraordinarily small numbers of participants, and it hasn't been peer-reviewed yet. As researchers are able to get more numbers and bigger amounts of data, we'll get a more nuanced view of what these mutations mean for reinfection rates, vaccine efficacy, how easily it is to transmit from person to person, and if it changes how sick people get with the virus. The last study I just wanted to talk about I thought was pretty interesting. The big challenge in trying to answer a lot of these very important salient questions is, well, we don't know. We're still in the thick of it, so we don't know what next year is going to bring, let alone next season, next month, next week. So this group of researchers tried to get clues about what could happen based on a different coronavirus. They picked coronavirus 229E. The reason they picked this coronavirus was for two particular reasons. One, we have established data on this going back 50 years. The second is the researchers felt that the spike protein, or how this binds, is very similar to SARS CoV-2. So they looked at 50 years of data and the antibodies that different people had at different stages of their life. Unsurprisingly, they found that the virus did change over years and decades. So what this group found was that as the virus changes, people's immunity to the virus changed. After somewhere between 8 and 17 years, the virus would mutate enough or change enough that your previous exposure, your previous infection, wouldn't provide much meaningful protection. So in some cases, coronavirus 229E had changed enough over that 10, 12, 15, 18 years that people who had previously been infected were susceptible again. And this is why we have an annual flu shot. It's more complicated with influenza in terms of why it changes year after year, but the idea is the same. It would be wonderful if after recovering from being infected with SARS CoV-2 or getting the vaccine, you were protected for over a decade. But it's just too early to know. And we're still rolling the vaccine out in the first place. And so trying to figure out what these various vaccines mean for transmissibility from person to person or duration of immunity is just going to take time. Previously, I mentioned that the United Kingdom has really led the global effort in doing genomic sequencing of the virus to find these mutations. Well, the CDC has been rolling out the NS3 program. The NS3 program is designed to continually be doing genomic sequencing across the United States. So between the CDC lab and collaborators all across the states, they're currently running 750 tests from all 50 states each week. On the CDC website, they note that the plan is to increase the number of genomic sequencing tests done to 1,750 per week. To date, the United States has done about 20% of the total genomic sequencing worldwide. That's just a little bit more than 50,000 sequencing tests. I would love it if we don't find any more mutations and we can figure out how the UK, the South African, and the Brazil strains differ, are similar, if certain therapies will work against certain strains but not others. But it seems likely that as we continue to go through this pandemic, that we will continue to find new strains. And what those given strains mean for all of us remains to be seen. But in general, my takeaways right now are it doesn't really change anything. And the preventative measures are going to be the same regardless of which strain is prevalent in your area. Wash your hands, wear the mask, social distance. Another question that I know many people are going to ask is, well, should I get the vaccine now or should I wait? Just get the vaccine. We went from having Chinese researchers release the entire genomic sequence of the virus in early 2020 to having a very effective vaccine by the end of 2020. If there does happen to be a very significant mutation that happens currently or in the future, I have the utmost confidence that those same scientists and researchers would be able to tweak existing vaccines to make them effective against multiple strains as they arise. So that'll do it for this episode. You know, the unknown is scary, and there have been an incredible amount of unknowns throughout the last year. But you should take comfort in the fact that there are a lot of researchers who are incredibly bright and incredibly smart and have incredible resources trying to figure out all these important questions, and will be until we're through the other end of this pandemic. So as new information comes to light, Lebowski reference, I'll present that to you. In the meantime, stay safe out there. Thanks for joining me, and I'll see you next time.